Okay, Assalamualaikum warahmatullahi wabarakatuh. Right, my name is Sharifah Faiza Said Muhammad, metric number 4191056. Today, I would like to present my individual assignment on Islamic philosophy regarding topic ethical issue of science and technology in the present world. And I specifically choose ethical issue related to human-animal hybrids, or also known as a chimera. So now let's go for the introduction. Okay, first we need to know what is chimera. Okay, we have a few definitions here. Okay, the first one is in Greek mythology. Okay, chimera describes a creature with a head of a lion, body of a goat, and a tail of a serpent. So here we can see three combination of animal in one individual, but quite different in modern medicine terminology. So in modern medicine, it describes a living organism that contains cells or tissue with different genotypes. Right? And in embryology, it refers to a combination of cells from different individuals. Okay, from two different individuals, we combine the cells together. And in molecular genetics, chimera is described as the combination of two DNA molecules from different individuals or from different chromosomes of the same individuals. Okay. And lastly, in genetic, chimera is described or refers to interspecies hybrids such as the mule, which is a cross of a female horse with a male donkey. So we have different definitions that describe what is a chimera, but basically, sorry, we can say that a chimera is a combination of two different individuals or two different subjects. We combine it into one. Okay. What and how they are produced? How is this chimera being produced? So, in a simpler words, chimera are animals that compose of cells that originate from two or more different species. Okay, and in the research lab, these are achieved by introducing cell of one species into the developing embryo or fetus of another species. Okay, so clear here, right? So these are example of a sheep-goat chimera. Okay, which is the first chimera created in 1984. Okay, it has a head of goat and the woolly coat of a sheep. Okay, now moving forward to the problem statement. First, the problem is about the production of human organs in animals, and then it generates a human-animal chimeras. So we involve human here, and lastly, these problems has followed by serious ethical issues. So we will discuss the issues later. Okay, now moving forward to the current project that utilize human-animal chimeras. So around the world, we have a few projects. There are a few projects that are ongoing. So the first one is the sheep-to-human organ transplant project. Okay, so this project is done at University of Nevada. Okay, where human stem cells were added to sheep fetuses to create chimera. Means that we take human stem cell and put it into the sheep fetuses in hoping it can serve as a reliable source of organ for liver transplant. Okay, some sheep may have 80% of human cells, and the sheep also have human cells in their hearts and their brain. Okay, so these are the findings from this project. Second project that is quite hot debate is the pig to human organ transplant project. So these are done in Mayo clinics in Minnesota. The there are a few risks doing this project. One is the risk of transmission of pig diseases to human. So we will discuss this later into detail. Okay, and the third project is mice with human brain cells. So this project were specifically objectively aimed to be used for study of neurologic diseases. For example, mice with human immune system that be used for AIDS drug testing. Okay, these are done in the Stanford University. They inject human neural stem cells into mouse fetuses. So the result was 1% of mice brains with human cells. Then they intended to increase until 100% like human brain in mice. Okay, so they will use this mice brain to study Parkinson and Alzheimer. Okay, so they have successfully developed a line of mice with human immune system, which are almost humans, and they are hoping to test new drugs of AIDS or HIV virus. Okay. So the fourth project, which is the rabbit human hybrids for stem cell research. So this is done at Shanghai Medical University. Okay, they develop embryos contain both humans and animal DNA. Okay, the goals of this project is to develop a new source of embryonic stem cells for medical research. Okay, last but not least, the project is monkey-human chimeras for study of Parkinson's disease. Okay. The scientists transplanting immature human brain cells into the brain of velvet monkeys, and their goal is to develop a treatment for Parkinson's disease. So we have looked at the current project that have utilizing human-animal chimera or animal-animal chimera. So these are referred to the webpage link that I've shown here. This one, okay. Okay, so how these animal and chimeric animals bearing humans organs. Okay, so these chimeric animals can be achieved because of the discovery of pluripotent cells. Okay, so these cells can differentiate into any cell types of organism, for instance, skin, liver, or heart. Okay, and pluripotency is a key property of a very specific human embryonic cell that has been found in an embryo stem cell. So basically, these cells are harvested from embryo, okay, embryonic stem cell, okay, and these cells are introduced into the embryo of another animals of different species. Okay. A second way to achieve chimeric animals is this one, chimeric animals by blastocyst complementation. So because of the discovery of pluripotent cell, scientists are able to inject the cells, the pluripotent cells, into the blastocyst of animals that we are desired to combine it. Okay. So this has created interspecific chimera animals and production of xenogenic organs. So looking back at this blastocyst complementation, for those that doesn't know what is a blastocyst is a structured form in the early developments of mammals. So after fertilization of eggs and sperm, it will become a blastocyst, zygotes, and then blastocyst. So this stage is prior to implementation of the embryo in mother's uterus, and it is about approximately 200 cells, and the embryonic stem cell that taken from one species is injected into a blastocyst of another species that will contribute to the embryo development. Okay, so we are clear about this blastocyst. So because of this, it leads to the interspecific chimerous animals and production of xenogenic organ. Okay. In 2010, a team of Hiromitsu Nakaude has reported that injection of rat-induced pluripotent stem cell into mouse blastocysts produce mice with organs that are included red cells. So, easier words, this research team has successfully injected of a rat iPSC. Okay, iPSC means induced pluripotent stem cell. So they take rat iPSC, this rat stem cell, injected into the embryo of a mice. Okay, embryo of a mouse. Okay, and then the results, they produce a mice with organs that contain the rat stem cell. Okay, so we have two species here, a rat and a mouse. Okay. So this same approach could be exploited to produce many other xenogenic organs in mice. Xenogenics mean different species organs we are using one organ from one species and transplant it into another species. Okay. Because of this, it will then leads to the production of human organs in animals. Okay, so these are the ultimate goal now. Try to produce human organs in animals, because previously this one, rat organs in animal can be produced in a mouse, leads to the idea of production of human organs in animal. And the question is, could it be possible to produce a xenogenic organ for humans in various animal species? Okay, so the carrier animal would be altered genetically in order to block development of a certain organs, and then the human-induced pluripotent stem cell will be injected into the blastocyst of that animals, of the altered genetically animal. Thus, only human cell would contribute to the development of the organs, and this animal will be raised until the human organ reach the required size, and then it will later be sacrificed before the transplantation date. So these are the ideas of producing human organs in animals. Okay? So among these chosen animals, okay, pigs is the on top of the list. So pigs is chosen, okay, because the size of their organs is similar to the size of human organs, okay, and second is because their metabolism, for example, their diet and temperature, is close to the human metabolism. This is highly debated using harvesting human organs inside the pigs animals container. Okay, so the pigs become as a surrogate or a container to harvest the human organ. So because of this, they have arise several ethical issues. The first one is about the retrovirus. So retrovirus is usually integrated in the genome of animals. So this virus can be transferred to human. So, but a few research of scientists has claimed that this transfer of retrovirus can be inactivated by using CRISPR or Cas9 technology. I'm not sure about this technology. If you want to know, you can search for yourself. Okay, so they have claimed that by applying this technology, the retrovirus transfer from animals to humans can be stopped. Okay, I think they are using the gene silencing technique. Okay, so second ethical issue is about the crossing the species boundaries. Okay, it is clear that we are using two different individuals, two different species here. So one animals and one human. So the animals that contain human tissue are called animals containing human material, or acronym as ACHM. So this has been the source of resistance by general publics. For example, the mice have been genetically modified to make them more susceptible to infection by human viruses, for example HIV, and human neuronal cells have been injected into the brain of the mice. Okay, so these are one example of different species boundaries. Okay, legal issues. So this human-animal chimera project also leads to legal issues. So it raises legal issues and is subjected to different framework in different countries. So later I will show you the table of rules and regulation in different countries. Okay. So the next one is the risk of development of human-like consciousness in chimeric animals. Okay. So this project has poses an ethical issue because uh, we need to differentiate between human and animals. Okay. So what makes us human? Will the injection of human iPSC into blastocysts humanize the animal brain? And then what differentiates us from animals? So basically, our brain have a capability to produce a consciousness. Okay, so this is ultimate differentiation between a human and animals. Okay, the ability of us to conscious and create emotion. Okay. So the next ethical issue is the risk of appearance of human-like external features. So scientists need to set a maximum threshold of human cell contribution in beforehand, and it is strictly implemented. Okay, scientists and researchers have to set a certain threshold limits of cell when they introduce the cell into the human. Right, and lastly is the risk of human gamete production. This is most likely not to occur, okay, because it is quite impossible to happen because the interspecies reproductive barrier is very strong. But cautions still need to be taken, okay, very not likely, okay, and then by, for example, introducing a suicide gene that would only be activated upon germinal differentiation. Okay, so these are among the ethical issues that integrated with the production of animal chimeras. Okay, so these are the table. Okay, from different countries, we have France, UK, Germany, USA, and Japan. Most of the countries are forbidden, allowed for a certain stage, but when they are into we're trying to further develops into the human, it is forbidden. But now Japan. Looking at Japan, currently limits an organ and human animal chimeric embryos. Okay, so by not allowing the development of such embryos beyond the appearance of the primitive trick, so they only allow the embryo to live until 14 days post-fertilization. But recently, the Japanese expert have supported the idea of creating a human-animal chimera and proposed that the Japanese research regulation should be capable of responding flexibly to the advance in research. So they are moving towards to allowing this research of human-animal chimeras in Japan. So I have a few examples here. So here I have a exclusive 2 pigs engineered to have monkey cell born in China. So these pigs contain monkey cells. This piglet had some cells from a monkey but died within a week of birth. Okay, they are successfully producing a piglet with a monkey cell inside it. Okay, and the second example here is a human pig chimeras embryo. This is done in 2017. Previously is 2019, which is very recent. Okay, and then this embryo were allowed to develop for 28 days. Okay, it contains less than 0.001% human, and the rest are pig. Okay. So what does Islamic say about this chimera project? So based on Ramli et al. 2014, it mentioned that interspecies chimeras, specifically the human-animal chimeras in stem cell research, was the most controversial issues in relation to human dignity. So we have an important issue here, which is human dignity. So according to Islamic sources of Al-Quran, Al-Hadith, and Ijtihad, there are 3 principles must be followed while conducting animal chimera research. The first one is the sources used in the research are not forbidden. I repeat, are not forbidden by the Sharia. Okay. Second, the product must be safe to the organ recipient. And the third, do not develop any kind of human dignity capacity. Okay, so these are the 3 important rules that have to be followed in order to in this research of chimeras. However, okay, we have some loose end here. If there is a conflict between human life and dignity, the protection of life is the primary concern. So we can choose life. more than dignity. However, this is a however, okay, but okay, we still have to access the dignity issue very detail of human animal chimera case by case with ultimate cautions. So this is what this paper have mentioned. And next, I will show you another paper that try to educating the Ummah by introducing Islamic bioethics in genetics and modern technology. So this paper has concluded that the main sources of Islamic law are Al-Quran and Sunnah. Okay, so other Islamic sources like Ijma' together with Qawaid Fiqhiyah and Maqasid Al-Sharia will be applied in order to frame the Islamic bioethics guidelines. So this is a very good paper. At least we as a scientist have a guideline, okay, has a guideline that we can follow in order to proceed with our experiments in whatever field that you are intended to go. Okay, so look at the Qawaid Fiqhiyah or Islamic legal maxim. This is actually rules, okay, basic rules of fiqh, okay, general rule of fiqh. Okay, it starts with first one, matters are determined according to intention. So whatever research that you are going to do is based on your intention. Okay. Second rules is hardship begets facility. So it means here that there are alternative and a way out which can be resorted in order to overcome difficulties and hardship. There always an alternative, okay, a solution to a problem. Okay. The third principle is harm should not be inflicted nor reciprocated to anyone or anything. All right. Fourth rule is what is certain cannot be removed by doubt. Means that anyway, whenever that you are feeling doubtful, you have to find a facts or evidence that will clear out your doubt. Okay. And lastly, custom is arbitrary. So this one refer to the general practices of the particular group or the practices of certain group of people, including the actions and saying. So whatever that particular groups normally do, we have to also refer to the general practices. Okay, so these are the 5 Qawaid Fiqhiyah. Now we're moving to the next one, is the objective of Sharia or Maqasid al-Sharia. This is also have discussed by another students in another topic, but the concept is similar. So we have 5 pillars here. The first pillar is preservation of religion and morality. Okay, so ibadah or worship such as salat, zakat, fasting and hajj are the elements for the maintenance of existence of ad-din. Okay, executing all this ritual will increase the iman of a person and therefore protecting the person from committing sins or preventing evil. Okay, so this is why very important. This is why we need to preserve religions and morality. Second pillar is the preservation of life and health. So we know that life is essential and valuable to everyone. So it is equally important and obligatory to each and every individual in society to sustain life, to preserve life. Okay. Third pillar is preservation of progeny. So this one is progeny is one of the essential elements of a person in his or her life. Okay, the distribution of wealth and prosperity, conservation of resources and sustainment of the environment is needed to ensure the generation of the next generation. Our next generation should be preserved, okay, not being harmed. The fourth pillar is preservation of intellect or mind. Okay, this one it specifically refers to an akal. Okay, our akal, our mind is a great gift from Allah to mankind. So this is one of the human capacities that differentiate man from animals. That's why we need to preserve intellect or mind. Okay, last but not least is the preservation of wealth or property. Okay, so we know that property is one of the necessities of mankind. So no one should transgress or take another one properties without the consent or legitimate reason and without proper contract. Okay, so we need to, so these are, okay, actually these are the 5 pillars of Maqasid al-Sharia. Okay, so with that, okay, I end my presentation. Okay, so I hope that I have share with you, okay, I have successfully shared with you a common problem regarding ethical issues on these chimeras or hybrid animals, and I hope you gain something from this presentation. Thank you very much for your time, and if you have any comments or question, you can leave behind at the comments section, and I really appreciate if you can click the like button to show your support. Okay, thank you. Assalamualaikum.